Okay. Yeah.
So good afternoon, everybody. We are going to start in a few seconds. Just take your seat. So I am Marco Candela. Maybe most of you already know me from the University of Bologna, Department of Pharmacy and Biotechnology. And I'm leading the group of microbiome science at biotechnology at the University of Bologna. So in the next few hours, we have 4 hours. We're going to treat some topics, so the arguments are quite wide, and we will face many things, and hopefully there will be something that would be interesting for you and also for your current research. So since I work on microbiology, I'm not sure everybody of you is familiar with microbiology. We will start from the basic principle of the microbial life. We will spend only a few minutes on that. Then we will see how microbiology changed with the multi-omic revolution. And so we will start to talk about microbiome, the microbiome science. What are microbiome? What is the microbiome science? Which are the main application of microbiome science in terms of discovery, but also opportunity of biotechnologies. And then we will see some technologies that are the basement of microbiome science. Mostly we will see next generation sequencing, how they work, and both the Illumina next generation sequencing technology, but also the third generation sequencing technology that is called long-read technology, such as PacBio, for instance. And then we will see also the differences and why both technology are important and somehow complementary. And then we will see a little bit of bioinformatics. So how the metagenomic data are analyzed, what we can do with metagenomic data up to the reads, assignment, assembly, and also the reconstruction of microbial genomes. And then at the end of the story, we will see a little bit of what we can do in terms of biodiscovery, exploiting metagenome in silico. So just using the available databases and the available software. So it's a kind of virtual biodiscovery that is nowadays it's becoming very important, because you will see there are a lot of opportunities just from these virtual approaches. And then there will be the homework, so the work that I will leave to you at home. That is a kind of virtual biodiscovery. So you will be you're going to use the available databases for biodiscovery, something a bioresources for the production of a biomolecule of interest for your application from your ecosystem. So we will see also these approaches, and then this is the basement of what you're going to do at home. Okay, if there are no questions, I would start with the first part. And so speaking about microbial life, and in particular, we will see the importance of the multi-omic revolution in terms of microbiology. So this is a very simple. So when we speak about microbiology, we speak about the invisible life. So we are speaking about fungi, bacteria, archaea, and virus are all microorganisms, and this is the life in the millimeter range. Okay, so this is microbiology. And maybe for sure you know how we can study microbiology. Of course, the first option is to visualize microorganisms by using microscope, but we can't understand anything about the microorganisms that we will visualize, just a visualization. The other option is to grow microorganisms in the lab and to make them visible. And then by growing the microorganisms in the lab and making them visible, we can start to study the microorganisms and understand which are their metabolic propensity and their functionalities. And the third option that is the most important today is that we can now study the microorganism just through the analysis of their molecular traces in the environment in which they live. And this is thanks to the multi-omics approach. So this is what I was saying about growing microorganisms. So as you know, a single microorganism is not visible. This is a very simple concept. So for make a microorganism visible and isolate the microorganism and microbial culture and being able to study the metabolic performance of a given microorganism, we need to be able to grow the microorganism in an agar plate and then make the microorganism visible as a colony, and then take a colony for exploring in the lab the biology of the microorganism. So this is very simple, is the culture-dependent microbiology. And everything since 15 years ago was dependent on the culture-dependent microbiology. Okay, everything was dependent on this. So let's see from an historical point of view, the microbiology and the technological innovation that allowed to change the paradigm in microbiology. So the first technological innovation was the invention of the microscope in 1670. And thanks to the invention of the microscope, it was possible to have for the first time the access to the invisible life. So this is the beginning of microbiology. There is something that is living, that is not visible at naked eyes, and we can see these microorganisms only thanks to the microbe. And then the second step was what we were talking about before, the invention of the cultivation-based approach to isolate microorganisms in single colony and make that visible, isolate the strain in the lab, so to be able to study their biochemistry and their functionalities. Okay, and this was so important and was invented in the middle of 1857. And the paradigm shift, the most important paradigm shift deriving from this invention was the Koch principle. So they have been able for the first time to isolate microorganisms for a sick animal and transmit the disease by the transmission of the microorganisms. So it was the first time the scientist was able to say, okay, microorganisms are the cause of the diseases for animal, for human beings, and for plants. But in a few years, the microbiology understood that microorganisms were not only the causes of diseases, but there were so many microorganisms that were important for nature. They were important for the biogeochemical cycle. And so we have, with the invention of the binographic column, the microbiology became able to isolate microorganisms even from environment, not only from diseases animal. And they understood that these microorganisms were important for the biology of Earth, of our planet. And then after 100 years, in 1983, it was invented for the first time, the PCR, the FISH, the Sanger sequencing, opening the way to the molecular microbiology, something that maybe you are all aware. So thanks to the molecular microbiology, I can relieve the presence of a given microorganism in a given sample by using some probes, some primers for the amplification of its DNA, some probes for the hybridization and for tailoring its DNA. And this is so important because it's allowed for the first time to detect the presence of a microorganism only in the natural environment without growing the microorganism, just detecting the presence of its DNA by specific probes. But the point is that for doing that, I need specific probes. I need specific primers tailoring the DNA of the given microorganism. I need a specific probe for the hybridization of the DNA of a given microorganism. And for creating specific primers and specific probes, I need to have the genomic DNA of the microorganism to be sequenced. This way I can design the probes. This way I can design the primers. And to have available the genomic DNA of a given microorganism, I need to have available the cell. So always, also in this case, the microbiology, this technology were dependent on the availability of isolates. And isolates are available only thanks to the application of culture-dependent approaches. Okay. So the point is that up to 2010, everything we knew of microbiology was limited to the fraction of the microbial life that were actually culturable in the lab. We didn't know anything about the microorganisms that were not actually culturable in the lab. Since it was not possible to study by culture-dependent approaches, it was not possible to isolate these microorganisms and create and sequence the DNA and develop probes of primers for their detection in the environment. Okay, so everything was limited to the cultural fraction of the microbial diversity. Only after the 2010, with the invention of the multi-omics approaches that are metagenomics, metatranscriptomics, and metabolomics, it became possible to study the microorganism in their native sample without any a priori hypothesis, and just see all the microorganisms that are there, that are living in a native sample, just characterizing their molecular traces. So the multi-omics approaches made the microbiology independent from the culture-dependent approaches. Okay, since it was possible to study the microbial life directly in the native environment, sequencing the entire DNA of the microorganism and characterizing all the molecules that were producing in the native environment. And so this opened the way to the culture-independent microbiology. And by applying the culture-independent microbiology, as microbiology, we become aware only 15 years ago that the cultural fraction of the microbial life is less than 2%. So up to 2010, everything we knew about the microbiology was limited to the 2% of the microbial life that was actually culturable in the lab. We didn't know anything about the rest. Now we know that there is the rest. Now we know that there is a huge diversity of microorganisms and microbial functionality that are important for life on Earth, thanks to our capacity to study the microbial life directly in the natural environment in target with untarget approaches, just sequencing and characterizing the molecular traces of these microorganisms by multi-omics technologies. So this was a complete revolution in microbiology, as you can imagine, since, of course, after 2010, the entire complexity of microbial life on planet became available to be studied, characterized, and exploited for biotechnology. So the first thing that was clear at that time was that in nature, microorganism lives in microbial community, where different microbial species interact. And this microbial community, where different microbial species interact, are called microbiomes. Okay, so microbiomes are everywhere, are in the air, in the water, of course, but in the soil, they live in association with the plant and live in association with animals. So there is no ecosystem, environmental or also associated on Earth, that is not populated by a microbiome. Okay, that is a functional part of the ecosystem, is part of the biodiversity of the ecosystem. So here's some definition about the microbiome science. So what is a microbiome? What is a microbiota? So starting from the microbiota. The microbiota is represented by all the living organisms that are part of living microorganisms, that are part of a microbial ecosystem. So given a microbial ecosystem, all the bacteria, archaea, fungi, protists, and algae that are living and lives in the ecosystem are represented microbiota. Okay, the microbiome is something different. Is the microbiota and the theater of activity. What I mean for the theater of the activity, all the microbial structural elements that are in an ecosystem because of the presence of the microbiota. So such as proteins, peptides, lipids, polysaccharides, nucleic acid, mobile genetic elements, and so on. Okay. And so this is something more. So it's the product from the microbiota that are released in the environment. But there is another very important thing that is part of the microbiome. that is the environment, are all the features that characterize the environment are part of the microbe, of the theater of activity. So there is the microbiota, what the microbiota release in the environment, but there is also the environment itself that is part of the microbe. And including, of course, all the metabolites that are released by the microbiota. So the toxin, the molecules, the signaling molecules that are released by the part of the microbiota. So there is the living part and all the products of the living parts and all the characteristics in terms of molecules and physiochemical characteristics of the environment in which the living part, the microbiota, lives. So how we can apply multi-omics to study microbiomes? Of course, we can run metagenomics approach. Metagenomics approach are functional to the characterization of the genomic DNA of all the microorganisms that are part of the microbiome. So all the microorganisms that are part of the microbiota. Okay. So by metagenomic application, we can understand which microorganisms are present in the microbiota and which is their functionalities. So what they are doing, what they can do. Okay. And then we can run metatranscriptomics by sequencing the RNA which is producing by these microorganisms. And by metatranscriptomic, we can understand what they are doing. So which gene they are transcribing while they live in an ecosystem, what they are doing. And then we can run metaproteomic. So okay, we can understand which kind of protein effector they are creating, which kind of proteins they are synthesizing, and how these proteins interact with the environment and with the biology of the single microorganism. And then finally, we can characterize all the metabolites that the microorganisms in the environment, that microorganisms produce in the environment by metabolomics application. So what they produce and how they change the environment by the provision of metabolites. So essentially, we can understand the complexity of the microbial life at different scale in terms of presence, functional potential, transcription, production of proteins, and production of metabolites. So we can figure out everything about microbial community and how this microbial community is interacting with the environment in which it's insisting and it's living. And so which is the role of the microbial community in the environment we found it lives. Sorry. So which are the main sources of microbiomes? So the major habitat are the subsurface. So the deep continental and the deep ocean subsurface. So microorganisms, living microorganisms have been identified up to 5 kilometers depth in the subsurface. And also another very important ecosystem is the sediment, the soil and the upper oceanic sediments, the marine waters as well. We will see a lot about the marine waters and the importance of this microbial ecosystem. And then there are all the host-associated ecosystems. So all the microbiomes that lives in association with specific hosts, such as herbivores, insects, mammalians, reptiles, and whatever, and also, of course, the plants. So essentially, when we speak about microbiome, we divide two types. We have two types of microbiomes. The one that lives in association with the complex hosts, such as animals, plants, and whatever, and the one that lives free in environment. So the first are the holobionts microbiome, the host-associated microbiomes. And the first are the environmental one. Okay, So what is important to know is that the host-associated microbiome is always a non-selected subset of the environmental microbial ecosystem. Okay, so they are always nested to the environmental microbial ecosystem. There is always a kind of connection. And what do? So the environmental microbiomes, so the one that lives in waters, air, soil, sediments, and whatever, and also subsurface, are essentially responsible for the biogeochemical cycles. So the recycling of nutrient between the organic and the mineral fraction that make life possible on Earth. And what about the host-associated microbiomes? The host-associated microbiomes are part of the physiology of the host in which they live in association. So they are important for the nutrition of the host. The plant and animal, doesn't matter. They are always important for the nutrition of the host. They are important for the defension of the host, for the interaction with the immune system of the host. And they provide essential physiological function that became part of the host biology. So we can see any complex organism on Earth by its own without considering the microbial counterpart. And so it's really clear now, thanks to the application of the metagenomic science, that microbiomes are somehow central from life on Earth, since they are important for living organisms. They are part of the living organisms, but they are also important for the biogeochemical cycling. So they are important for nutrient recycling on Earth to make life possible on our planet. So this this made has been this resulted in what is called in a theory that is called the microbiome centrality. So the point is that microbial ecosystem in their specificity and differentiation are the life support system of the global biosphere. So whatever they are, host-associated and environmental, they always provide key ecological service to support the planet health. Just think about that the 50% of the oxygen production on the planet is due to the microbiomes. All the carbon and nutrient cycling is due to the microbiome. So they are the basement of the global food webs. And all holobiomes, so plants and animals, can live on our planet only thanks to the symbiosis with microbiomes. So this is the microbiome central. So there is no life in the planet without microbiomes. Since microbiome made the planet habitable, but also allow complex organisms to live on our planet. So that's to say that microbiomes are obviously central in the One Earth theory. So if we see, we provide an integrated vision of the human health, environmental health, and animal health, and whatever, of course, microbiomes are part of the One Earth theory. So all these health aspects involve their own microbiomes and the interaction between their own microbiomes and the microbial ecosystem on the environment. Okay, so they are central for the One Health theory. So what about the microbiome biotechnology? So the microbiome biotechnology is based on the concept that once we understand the complex functionalities that microbial ecosystem can do, we can try to exploit these functionalities to solve problems by running out biotechnological applications. So, and this is the argument of the microbiome research and innovation. So if we understand better how microbiomes works, in providing all these ecosystem service in support of life in the planets, we can apply some microbiome-based solution for resolving sustainable development goals. So we can hit many sustainable development goals by using microbiomes. We will see as a source of biotechnological solution. And this is the goal of the microbiome science. So the microbiome science essentially starts from the biology. That means study microbial ecosystem, so the biological part of the microbial ecosystem or the host, but involve also chemistry, since we need to know how the microorganisms interact with an host or interact with an ecosystem by exchanging molecules and transforming molecules and make reaction. But also needs mathematics, since we need to apply informatics, statistics, and modeling to study microbial ecosystem. Because informatics is important, since multi-omics are highly connected to informatics and statistics, because multi-omics provides a huge multidimensional data set that needs informatics, statistics, and modelings to be analyzed. But also, once we understand, thanks to biology, chemistry, and mathematics, how microbiomes works, we need biotechnology for using for deriving application in different fields from what we have learned by studying microbial ecosystem by biotechnological application, by multi-omics application. And the application are in different fields, such as medicine, animal husbandry, agronomy, bioeconomy, and food science. We will see some example of microbiome application of the microbiome-based science and biotechnology in all these fields of research. This is a cover of a Nature journal from a few years ago that was totally dedicated to what is called the miraculous microbiome. So the fact that by using microbiomes, we can provide solution. And this is something that you will learn how it's possible to do today. Okay. And why microbiomes? Why microbiomes are so important for providing solution, biotechnological solution in terms of achievement of the sustainable development goals? Because essentially, if we think about life on Earth, bacteria represent the 60% of the biochemically active biomass. So the world microbial biomass essentially is the main catalyzer of biochemical reaction on Earth. So the 60% of the biomass that is catalyzing it, bioactive, is microbial, it's microbiome. So they are responsible of quite all the biotransformation that we have on our planet. Okay, so the microbiome is the most important genetically determined catalysis that we have on our planet. And this is so important for carbon upcycling. That means create high-value compound from waste, or one carbon biotechnology. That means create deliverable, marketable items from CO2 capture or carbon dioxide capture. Okay, so it's bioactive, biodiverse component that we need to exploit for sustainable application, because it's the microbiomes on Earth represent a huge bioactive, genetically diverse system that we can exploit for usable biotransformation for the transition to a sustainable planet. And this, for example, is the importance they have for in terms of carbon fixation. So microbiomes are strategic in the carbon dioxide fixation, since they have different systems they can exploit for fixing carbon, not only in the presence of oxygen, but also in an anaerobic condition. And here there is various publication on Cell, in which there is, it's quite interesting, the direct connection of the most important functionalities that can be provided by microbiomes, whatever they are, environmental or host-associated, in terms of application by providing by technological application to achieve sustainable development goals. Okay, so here you have the functionalities, and here you have the sustainable development goals you can achieve by exploiting these microbiome-dependent functionalities. And so essentially it's believed that the 60% of the inputs to the global economy can be produced by microbiomes. In terms of what? In terms of pharmaceutical, new pharmaceutical, or new nutraceutical, synthetic chemicals for the food system, food preservatives, for instance, protein, fats, carbohydrate as a nutrient, food, and polymers that can be used, biomaterials such as the bioplastics, or fuels such as butanol, biological oils, hydrogens, can all be produced by microbiomes. So you see many different items that are so important in the circular economy for the transition to sustainable development, to sustainable world by eating sustainable development goals can be provided by microbial ecosystem. So this slide is to convince you how it is important to study microbiomes, to understand what they can do to find solution in terms of biotechnological application to achieve the sustainable development goals by means of circular process. Okay, and the exploring, this kind of exploration in terms of microbiome structure for finding solution in that term is of course called microbiome biodiscovery. So search for feature in this microbial ecosystem, whatever they are, for solving solution. And so we will see now what we need for microbiome biodiscovery in terms of technology and how we can run this microbiome biodiscovery in silico without the need of a lab. So the next point is metagenomics. So we're going to speak a little bit about metagenomics. That is the technology, that we need to apply to study microbial ecosystem, to sequence their DNA. Okay. So metagenomics, it's merely the untarget sequencing of the genetic material that is present in a given sample, nothing more. So you get a sample from the environment, you extract the DNA, you sequence and target all the DNA that is present in a sample, and this is metagenomics, nothing more. Okay, so it's very easy. And when was invented metagenomics? Metagenomics was invented in 2004 by Greg Venter. This is the first publication about metagenomic science. And you know who is Greg Venter? Some of you knows. Greg Venter is the first person that was successful in sequencing the human genome. When? In 2004. Same here. So the same here was sequencing the human genome and providing the first application of metagenomic science. And the Greg Venter at that time, in 2004, he was at the university. And then he got out from the university because he didn't like the university system from the United States, of course. And it was Stanford, and he created its own institution, the Greg Venter Institute. There is already. The Great Barrier Institute is already active. You can Google and you will see what they are doing. Okay, and with the Great Barrier Institute, it was challenging all the other academic institutions on our planet for sequencing first the human genome. So at that time, there were all the universities in the planet against the Great Barrier Institute for sequencing the human genome. So it was a race, and Great Barrier won. So it was the first to sequence the human genome before a huge consortium with all the other universities, United States and Europe on the planet. It was crazy. And you know, which is the first genome, human genome, have been sequenced? Just guess? Yeah. His genome. So he is also the first human genome being sequenced. So the reference is Great Barrier genome. So anyway, we are speaking about metagenome. It is the same thing with metagenome application. So it was the first to think that, well, yeah, it's possible. That I try to get all the DNA that is present in the sample, and without any a priori hypothesis, I sequence everything. And what he was discovering, that it was sequencing just 5 samples he collected in the Sargasso Sea. So just 5, and it was publishing in Science. So now to publish in Science, you need 500 samples, 5,000 samples. So with 5 samples, it's not possible. And what he was discovering by running a metagenomic application, just sequencing the DNA collected from these random samples, water samples in the Sargasso Sea, he discovered that there were many more microorganisms that we were expecting to see. So he discovered for the first time the part of the microbial life that is not culturable. Okay, with the first metagenomic application, where we were expecting to find 10 microorganisms, 10 different species, there were actually 1,000 different species. Because only 10 are culturable. The rest are unculturable and can be seen only thanks to metagenomic application. And this is the only discovery he did, nothing else. Just to say, oh guys, there are many microorganisms that we were expect to see by applying the standard culture-dependent technologies. There are 1,000 times more. So the microbiology is more complex. There is more microorganisms, more functionalities, more diversity that we expected to see. So, but in 2004, running a metagenomic application was still dependent on Sanger sequencing technology. Sanger sequencing technology is the standard technology for DNA sequencing. So it's expensive and time consuming. So for sequencing by metagenomic application, only 4 samples, it was taking years, for getting this data by running Sanger sequencing technology. So the metagenomic application, even if at that time providing information of a great potentiality, so informing about the real complexity and diversity of microbial worlds to be exploited, is what's not applicable, since by Sanger sequencing, the performance of the sequencing was too low to be applied at larger scale in metagenomic study. So indeed, nothing happened for 10 years. So it was just a principle. And after 10 years, it was invented a new approach to sequence, that is next generation sequencing, that made sequencing technology more performative. So they became cheaper to sequence. And this technology were providing a lot of more data with respect to the Sanger technology. So it was perfect for metagenomic application. So if I ran a discipline that is tailored to the sequencing of DNA, whatever it is, I need a technology for sequencing that is high performative, provide a lot of sequence base with a low cost. Okay. Otherwise, I can run this type of metagenomic application. And this is what happened with next generation sequencing in 2010 and later on. So let's talk about a little bit next generation sequencing. What we can do with next generation sequencing is that it's simple. We have a microbial community. Here you have different taxa, different color and different shape. And from a microbial community, we can extract the DNA. And then by running next generation sequencing, so metagenomic application with next generation sequencing approach, we can sequence this DNA. We can sequence the entire DNA we have and getting the information in terms of microorganisms that are present there and also the functionalities that are characteristic for these microorganisms. But we can also limit our sequencing to a single gene for a given copy of a given genome type. If that is marker, that gene is a phylogenetic marker, by sequencing the phylogenetic marker for even copy of genomes for each genome types, we can understand which microorganisms are present in the ecosystem. Okay, not the function, since we are not sequencing the entire genome. So there are two applications. One is metagenomics. In terms, I sequence all the genomes of the microorganisms present in an ecosystem, and the other is a marker gene approach, in which I sequence only the phylogenetic markers for every copy of genomes present in a microbial ecosystem. Okay. And the third application is metatranscriptomic, that is the sequencing of the RNA, which is produced by a microbial community. And from the technological point of view, it's exactly as sequencing the genomic DNA. Since from the RNA, you create a cDNA, and then you sequence the cDNA, as it would be the genomic DNA. Okay. So what we will see about technologies for sequencing the genomes in terms of metagenomes is the same for sequencing the transcriptome. So which is the marker gene? The marker gene that is mostly used for microbiology application is the 16S38 gene. Since it's recognized phylogenetic marker, since this gene is characterized by some conserved regions that are the same for each microorganism on planet, and some hypervariable regions that has a species-specific features in terms of sequence. Okay, so this is the best phylogenetic clock and also marker gene for the phylogenetic reconstructor that is used in marker gene approach for metagenomic application nowadays. So we won't talk a lot about this because I'm much more interested in the metagenome sequencing, so the sequencing of the entire genomes. Okay, that is most important for the bioprospecting and also for future application. Okay, since of course is more informative. And so let's see how, which is the process, starting from the sample, the native samples, up to the sequencing and up to the bioinformatics for a metagenomic study. So first of all, we need to start with the collection of a microbiome sample. What is a microbiome sample? Is a native sample corresponding to its theater of activity. So nothing more. So if I'm studying the soil microbiome, a sample of the soil microbiome is a sample of soil. If I'm studying the water microbiome, the sample of the water microbiome, marine water, is the sample of water. If I'm studying the air, the sample of air. If I'm studying the microbiome is associated with a plant, a sample of the plant associated microbiome is a sample of the rhizosphere. Okay, so it's always a sample of the native ecosystem that correspond to the theater of activity of a given microbe. Okay, the only important point is that that sample needs to contain enough DNA for running a next generation sequencing reaction, for being able to sequence the metagenome. Okay, so the sample collection is theoretically very easy. So just collect the natural sample. But the point is that you need to do that with validated approaches. The validated approach means that at least you need to be sure that you collect enough theater of activity to collect enough DNA for running a sequencing reaction. And also you need to be sure that from the collection to the lab, nothing changed in your sample. So you need to preserve the sample up to the delivery to the lab. And you can do that, for instance, there are several approaches for preservation of the sample. One is freezing, if possible. The other is desiccation, or you can use preservation buffer. So you get the native sample. You need to be sure that in the native sample there is enough DNA for running a sequencing. Then you need to apply a preservation measure, freezing, desiccation, or using a buffer up to the time the sample reaches the lab. Okay. And when you're in lab, you start with the DNA extraction. DNA extraction is very easy. There are so many kits for DNA extraction. It's a matter of 1 hour, 1 hour and a half. And you can get the DNA from any kind of samples, just using commercial kit. And when you get the DNA, what do you do? When you get it, you need to check for the quality of the DNA. You need to check for the quantity, for the contamination, and for the integrity of the DNA you have extracted. Only if the DNA is not with good quality, low contamination, and not degraded, you can run the sequencing reaction. Okay. So of course there are many different instruments for doing that by using speedometer or using robot for running capillary electrophoresis for testing the DNA quality in different samples at the same time. Okay. So if you got a sample, you have been successful in the extraction of the DNA. It's of good quality, not contaminated. It's enough for running a sequencing reaction. What you have to do now is to prepare the DNA for the sequencing. So it's called the library preparation. Okay. So there are two sequencing technologies. There are more, but the most important are that two. So when you have DNA, you can choose two technologies for sequencing. The one is the short read technologies that the leading company providing this technology is Illumina. And it's characterized by the fact that it provides a lot of base sequences. So it's able to sequence a lot of bases, so up to 3 terabases, but in short fragments. So the short sequencing technology provides a lot of sequence, so a lot of base, but it's able to sequence only 100, 300 base pair fragments, no more. Okay. The other technology that is the long read technology that is called PacBio has different performance. So it can provide only 20 gigabase of DNA sequenced for each run, so less DNA, so 1,000 less DNA, but in longer fragments. So the fragments can be up to 30 KB. 30,000 bases. Okay, this is a different approach. It's quite complementary. So with the Lumina, you can go very deep in sequencing. You can sequence a lot of DNA. So you can get the entire biodiversity in a sample. But then you have short fragments. And with the short fragments, it's much more complicated to assign the functionality and to reconstruct the genomes. With the long read technology, you sequence less diversity. But, yeah, we will see. No, this is another important difference. We will see when we will discuss the two technology. We will see the difference. Yeah, and it's PCR independent. So this is why also PacBio make less errors. So because it sequences directly the single DNA molecules, it does not depend on PCR. We will see. And so there are these two technologies, which is the best, both. So the better would be have available two approaches. So at first, you start with the shotgun Illumina sequencing for sequencing the entire diversity of the ecosystem, and then with Rodrigo technology for refining sequencing for the most abundant microorganisms, for instance, but only to extract very precise information and to close the genomes of the most abundant ones. So they are quite complementary. So if you need to choose one, I would choose Illumina, since it's with Illumina, you can try to do everything. So even to study the genomes, but also to eat the entire diversity, the entire biodiversity. With the first one, it's limited in terms of the diversity you can catch. So let's see the principle. So we don't spend so much time in discussing how the sequencing technology works because it needs too much time. So the principle is that of Illumina sequencing. This is a metagenome. What does mean a metagenome? This comes from a microbiota, from which imagine you have three types of microorganisms. Okay, the blue microorganism, the red microorganism, and the black microorganism. Each of these microorganisms has its own genome. So when you extract the DNA, you have the genomes from the blue microorganisms, the genomes from the red microorganisms, and the genomes from the black microorganisms. Okay? And so this is the metagenome. What is important from a metagenome is that the abundance of a genome type, the blue genome type, is proportional to the abundance of the blue microorganism. Okay, the abundance of the red genome type is proportional to the abundance of the red microorganism in the microbiome. So the abundance of a given genome type is abundance in terms of number of genome copy is proportional to the abundance of microorganisms providing that genome type. Okay, so this is the metagenome that mirror the microbiota. Abundance of genomes type is exactly the abundance of the different species type. And then with the metagenome, the first things you have to do for running a Illumina sequencing is a random fragmentation. This is so important, the random fragmentation of any copy of genome per genome type in the metagenome. So each one is fragmented in fragments of 300 base pair in a random process. So from the genome blue, from the 4 copy of genome blue here, you would obtain a number of random fragments. Okay. So at the end of the story, you have a population of random fragments obtained from the fragmentation of the different copies of each genome type in the metagenome that is proportional to the number of microorganisms belonging to each species providing its genome type. Okay. So you have much more random fragments from the most abundant blue microorganisms for the most abundant blue genome, and less fragments from the red microorganisms. Okay. So there is always this proportionality. The abundance of a species, abundance of a genome, abundance of fragments obtained from the random fragmentation of that genomes. Okay. So how you run the random fragmentation? So imagine you have your sample. Imagine water sample, marine water. You extract the DNA from the marine water, and then you have the metagenome with all the different genome copy per genome type. What you do with that metagenome, you need to start with that random fragmentation. Okay, so this is a very important step. And how you can do the random fragmentation? By using some transposon, and in a process that is called fragmentation. You see here, you know, what are transposon? Transposon are proteins that cut the DNA randomly every 300 base pair. Okay. And so by using this transposon, you have the random fragmentation of any genome copy per any genome type by using such kind of enzyme. Okay. And the fragments that you get are of 300 base pair. Okay. So here, just see the fragmentation process. What is important is that this transposon, while they cleave the DNA, they also add some adapters at the end of each fragment. Okay, look at here. This is a specific DNA molecule. They cut the molecule in a fragments of 300 base pair, and at the 5 first of the fragments, left side, there is a blue adapter that is a synthetic DNA molecule. Okay, and at the right side of the fragments, there is a green adapter that is another synthetic DNA molecule. Okay. So this way, by using the fragmentation, you are able to fragment any copy of DNA in random fragments of 300 base pairs. At both sides of each fragment, you have two adapters. The blue one at the left side, the green one at the right side. Okay. And then what you do? You exploit this adapter for what is called the index PCR. So these are all the fragments you get. Okay, so imagine millions of fragments obtained from the fragmentation of the many genome copies from a given metagenomes, and then you amplify these fragments with these two primers. The left primers as a region that is homologous to the adapters you have included by the fragmentation. That is also the read sequencing primer one. And then you have an index for barcoding the fragments, and then you have a P5 reason for the amplification, for the bridge amplification, you will see. And here is the right primer. You have read the sequencing primer to an index to a P7 region. Okay. So at the end of the story, every fragment, so every insert from your metagenomes, so I'm speaking about millions of fragments of 300 base pair obtained from the fragmentation of the so many copy of different microbial DNA you have in a metagenome, a microbiome sample. Every fragment is in between two huge adapters. Okay. So here you have the fragments that is called insert, that is the unknown sequence, it is the unknown DNA, so it's a fragment from the genome. And then you have the adapters. In the adapter, you have the read sequencing primer 1 and the read sequencing primer 2 that are necessary for running a sequencing reaction. And then you have an index here that is a DNA barcode for recognizing all the fragments derived from the fragmentation of the given DNA sample, so a given metagenome sample. And then you have these two reasons, P5 and P7, okay, that are functional for the sequencing reaction. So how sequencing works? Sequencing in Illumina sequencing, it's exploit a technology that is called surface sequencing reaction. So this is the fragment, is the same, okay? And here you have the insert, and this is one of the billion of different fragments you obtain from the library preparation from a single metagenome sample, okay? And so all the fragments are the same for the adapters, but the differences are the insert, okay? The fragment of the native DNA sample, okay? And as we say, the adapters, in the adapters, you have the read sequencing primer 1, a barcode, and the P5 and P7 region. Okay, so let's see what happened. Just imagine that you denaturate this double strand into single strand. Okay, and imagine that you have the forward strand here and the reverse strand. Okay, imagine that you can immobilize each strand by the 5 first on a surface. Okay, so look at this strand and see that is immobilized here at the 5 first on a given surface. And the same happened to the right strand. The right strand is immobilized at the P7 region on a surface. Okay, now you have the two single strands that are immobilized on a surface. And now, with these two single strands, you can start sequencing. You start just to get the sequence of the forward strand. How? Sequencing from the read sequencing primer 1, the reverse strand. Okay, so just polymerizing from the read sequencing primer 1, the forward strand, copying the reverse strand, you get the sequence of the forward, okay? And on the other side, by sequencing from the read sequencing primer 2 on the forward strand, you copy the forward strand, generating the reverse strand. And while you generate the new strand, you also sequence, okay? This is the basement of the sequencing reaction, okay? So essentially, by using these two adapters, you put the insert in between two adapters that allow the primers for sequencing the DNA of the insert to be sequenced. Okay, you get this? Okay, which is the problem with the example? So imagine that here you have the plate. Okay, so it's not working so well. Okay. And then here you have a fragment. Okay, with the two adapters. Okay, just one. These fragments with the two adapters hybridize on a given side on the plate here. Okay. And then you can run the sequence of the insert by using the two primers that are in the adapters, as I was saying. Before doing that, and this is the story of a fragment, okay? Before doing that, you need to amplify the fragments. So to run a PCR reaction on site on the flow cell to obtain a clone copy of the fragments. This is functional to amplify the sequence. Okay. So with the fragments, you hybridize the fragments on a specific region of the flow cell. Then you run a PCR to obtain many copies of the same fragments. Okay. That is called a sequencing cluster. And then you sequence all the copies at the same time. This is needed to amplify the signal of sequencing. Okay. So that's to explain you that sequencing is dependent on PCR. And PCR happen on the surface that is called flow cell. Okay. So the entire story is that you have a microbial genome, like just one. It's imagine the same from many copy of different genomes type in different number in different manner. Okay, so and you obtain fragments. Okay, so many fragments, millions of fragments. Each fragment is in between two adapters, in which there are the primers for sequencing reaction. Okay, then you need to let each fragment find a place in the flow cell. You have these fragments would be here, and these fragments would be here. Okay. And then after the fragments find a place in the flow cell, you need to amplify locally the fragments to create a sequencing cluster. Okay. So you would have the amplification here. And you create a cluster, right? This one. And now you can sequence the cluster. You sequence at the same time many identical copy of the same insert. Okay. But here you can create errors because you amplify the cluster. Okay. So this is how it works. So it's strictly dependent on the amplification of the fragments on the flow cell. So let's see another very important point. So the difference between sequencing a single genome and sequencing a metagenome. It's a completely different story. Just imagine here you have a single genome, okay? And just imagine that you have 245 megabase is the dimension of a single genome, okay? So as a good practice, to be sure about your sequencing, the genome sequencing, you need to sequence at least 10 times every base of the genome, okay? So for doing that, it's easy. You need at least 10 genomes, copies from the genome type blue, okay? This way, you can sequence 1 time each copy, and you have sequenced 10 times in total the genome. Okay, so it's very easy to get 10 genome copies of a single genomes of a microorganism. So it's matter to have 10 microorganisms. It's very few. Okay, it's not a problem. So what you can do? So just imagine that you start with 200.5 megabase genome. So you have 10 copies of 200.5 megabase genome. Okay, I understand. Then you start with the random fragmentation, as we have seen. And after the random fragmentation, you need to add the adapter for the sequencing with the read sequencing primer. Okay. And so, how many fragments you get? You get 100,300 base pair fragments. That is the result of the fragmentation of 10 genome copies, each of 200 base pair, 200.5 megabase. Okay. So just imagine that you get 10,300 base pair fragments per genome copy. That is 2.5 megabase divided for 300 base pair. And of course, each genome copies provide 10,300 base pair fragments. So it's 100,300 base pair fragments. Okay. And then you need to find a position in the flow cell for creating a sequencing cluster for each of these fragments. Okay. So you have 10 genome copies. You have all the fragments obtained from the 10 genome copies, that all the fragments are in between the two inserts, and then to be sequenced, all the fragments need to create a cluster, a sequencing clusters by clonal amplification, as I was saying before, in a specific region of the flow cell. You agree? So for sequencing a single genome with good coverage, you need to be able to sequence 100,300 base pair fragments, and you need to be able to have available on your flow cell 100,000 sequencing clusters. Okay. With 100,000 sequencing cluster, you have enough sequencing power for sequence 100,300 base pair fragments that are enough for sequencing all the fragments that you generate from 10 copies of a microbial genome. But what happened with the metagenome? Consider that in a metagenome, this is a very simplified version of a metagenome, okay? In which you have a minor component that is a major component. The major component is the red microorganism, is 10 times more abundant than the minor component, the blue one, okay? And they are together in the same environment. That means that when you extract the DNA, the resulting DNA from this population is diluted in the same solution. Okay, so you will get a metagenome DNA as a solution of DNA in which there is always a genome type that is 10 times more abundant than the other one. Okay, since in the microbiome, the different microorganisms are not always in the same abundance as we have seen before. You have the dominant and the subdominant. It's very easy that there is a guy that is 10 times more abundant than the other one, and so providing 10 times more copy of bacterial genome. Okay, so let's try to sequence this metagenome. Okay, so as you know, we have gold standard. So for sequencing the microbial genomes from the two microorganisms, you need to sequence the fragments generated by the fragmentation of at least 10 copies of microbial genomes of a given microorganism. So if you get enough the solution in which you have 10 genome copies of this less abundant microorganism, that is a minimal amount, for getting 100,300 base fragments for sequencing the fragments resulting from the 10 genome copies. In the same solution, you will have also the 100 genome copy from the most abundant one, since they are together in the solution, generating 1 million of 300 base pair fragments. Okay. So when you sequence a metagenome, for sequencing with the good coverage, all the fragments generated for the fragmentation of the 10 genome copies of the less abundant microorganisms, you need also to sequence the millions of fragments generated from the co-fragmentation of the 100 genome copies of the microorganism that is 10 times more abundant. So you need 1 million of sequencing clusters to sequence all these fragments. And you will sequence with 10 times coverage the less abundant genome, and with 100 times coverage the most abundant one. Okay, this is why for metagenomic application you need this huge sequencing power, because to sequence the less abundant target, you would sequence always a lot of DNA coming from the most abundant one. Okay, so you need a machine that is able to create 1 million sequencing clusters, each one generating the reads from an insert, mostly from the most abundant genome type, some of the less abundant genome type. Okay, so you understand the difference in terms of sequencing power needed for sequencing a single genome or sequencing a metagenome. Okay, you need a lot of sequencing power. And which is the point that when you sequence a metagenome, you doesn't know how many microorganisms there are. You doesn't know which is the dimension of their genomes. And you doesn't know the differences in terms of relative abundances. You don't know anything. So what you can do, how you can start, which is the right number of sequencing cluster you need to generate for sequencing a metagenome. So which is the right number of sequencing read, since each sequencing cluster provides a read sequence. You need to have available for sequencing a metagenome. You never know. You need to try and see how much diversity you have been able to get. So, of course, there are good practice, nothing else. So for a single shotgun metagenome sequencing, for single samples, you need to provide between 5 to 100 million reads. So that means that you need to provide between 5 to 100 million sequencing clusters, each sequencing a given fragments obtained from the fragmentation of the metagenome. Okay. So if you, when you will see some papers, you will see the depth of sequencing. If it's shotgun metagenomics, it won't be less than 5 million reads. Okay. Otherwise, it's shallow sequencing. You have sequencing only the most abundant microorganisms in the microbiome. Okay. So these are the Illumina instruments you have available and the performances. You see, it's very easy. Of course, there are many systems, and the difference is the performance in terms of millions of reads. Okay, that is the same to say in terms of millions of clusters they can generate for providing the read of a given insert after the clonal amplification of the Illumina surface. Okay, and so see here the less performative provide 25 billions of reads. That means that you can sequence only few metagenome by using this instrument. Here you have the next one that provide 40,000, 400 billions of reads. And here you can start, you can able to sequence, let's say 20 up to 50 metagenome. And then you pay more and you have the highest performance up to 5 billion, 6 billions and 20 billions of reads. in which you can sequence thousands of metagenome in the same run. Okay. So for sequencing thousands of metagenome in the same run, the only thing you need to do is to barcode the fragments obtained from the fragmentation of each metagenome in a different way. So you remember we have seen very fast the fragmentation and then amplification for the barcoding. So the addition on both sides of the fragments of the two adapters. Okay. So, of course, you can use different combination of barcodes with different samples. So you have the metagenome number 1 with its own complexity. You fragment the molecules, and then you, by PCR, you add the adapters, including a specific barcode from all the fragments that generated by this sample. And here is represented as purple. Okay. And then we have the second sample, you fragmentation, and then the addition of the adapters with the barcode green. The fragmentation and the addition of the adapters with the barcode yellow. A barcode is just a specific DNA sequence by which you can tag any molecules that comes from the fragmentation of a given sample. This way, if all the fragments generated from all the samples are barcoded in between two specific artificial and recognizable DNA sequence, then you can put the fragments together, sequence all the fragments together as if they would be part of the same huge metagenome. And then, even if as you are sequencing, then basing on the barcodes, you can separate the reads according to the sample of origin. So when you sequence, all the reads, sequencing reads that are connected with the barcode purple are separated in a single file. All the reads that are connected with the barcode yellow are separated in another single file. So this way, you can sequence many samples at the same time and recognize the sequence belonging to each metagenome just because they are all tagged with a specific barcode. And so we can make a break now of 5 minutes, and then we will see the PacBio. 10, as you want. 10. Yeah. Tu sei proprio un po' là.
Sento appena adesso sono io sempre qua.
Dopo mi risento.
Un attimo il microfono per...
specialista lo chiuso, ma le sue cose science, per trasportare queste cose che abbiamo con le varie articoli nostri, pensavo una ventina di 10 articoli, sono arrivati 37, sono arrivati a scienza. Perché poi mi tocca rileggere tutte le risposte ai migliori review per capire se sono le capito? Allora si può dietro questa settimana la parte dietro.
Ok, grazie. Quanto avete fatto in questo bicchiere? Okay, so can go on. So now it's a time of the third generation sequencing approach, and in particular we will speak about the PacBio technology. So the point is that as we have seen, the second generation sequencing approach, so Illumina sequencing, needs clonal amplification. So every fragments we obtain from the fragmentation of the original metagenome, it's amplified before the sequencing reaction to allow to amplify the signal of the sequencing reaction, which is functional to the sequencing itself. So, of course, when you amplify the DNA, the point is that you also add some possible errors, and this is the limit. So, the point is that since second sequencing reaction is dependent on the clonal amplification, that makes some risk in terms of error of sequencing. The other point is that, as we have seen, the Illumina sequencing provides sequence only of 100 base pair, 300 base pair, no more. So it's able to provide only short fragments. And this makes difficulties when you want to assign functionality and also when you want to assemble the genome from this very short. So the third generation sequencing was invented to face this problem. So to sequence without the need of a clonal amplification for the sequencing reaction, and also to be able to sequence longer fragments for allowing an easiest reconstruction of bacterial genomes from the metagenome, but also a much more trustable assignment of the sequence functions. So the leading technology in terms of third generation sequencing is this one. It's called PacBio, and it has been conceived to sequence a single DNA molecule in real time. Okay. Indeed, the technology, it's called single molecule real-time sequencing. So, doesn't need clonal amplification, but by using this approach, you can sequence directly the original fragments generated from the original metagenome. There is no PCR in between. So no error due to the PCR reaction. So how it works? First of all, you generate fragments from the metagenome. The difference is that the fragments now are not of 300 base pair, but are of 30,000 base pair. Okay, so it's 30 kilobase. So it's a longer fragment. Then after the generation of the fragment, this is a fragment from the native metagenome, you add this kind of structure at the very end of the fragments. You see, it's a kind of loop. Okay, and this is a reaction which is provided by the backbio. So it's me just running the protocol. And by the addition of this loop, essentially you circularize the double strand. Just imagine to open this double strand, you create something like a plasmid, okay? And then, so with the circular double strand, you can sequence directly the fragments. Of course, you would sequence first the reverse strand, generating the sequence to the forward, then the loop, then you sequence the forward strand, generating the sequence of the reverse strand, then the loop, then you sequence again the reverse strand, generating the forward. So you can sequence more time, the same fragments, both left strand and right strand in a circular process. Okay. So the other point is that by doing so, you can generate more sequence from the same fragment, from the same native fragments, to be really sure you don't commit error. Okay. So the other very important point of this technology is that the DNA polymerase, so the enzyme that is needed for sequencing, is somehow connected at the here at the depth of a single well. So there are, just imagine that you have a kind of flow cell in which you have many wells. At the bottom of each well, so they are just 100 nanometer in diameter. At the end of each well here, you have a single DNA polymerase molecule which is bounded at the bottom of the well. Okay. Then, just controlling for dilution, you make it possible that only a single fragment with the loop enter within each well. And in each well, the DNA polymerase starts sequencing the loop. Okay. And so from each well, you have the signal of a light signal that is responsible, is corresponding to the single nucleotides that are added time after time by the DNA polymerase while sequencing the single DNA molecule. Okay. So each well is a sequencing reactor with a DNA polymerase anchored at the very end of the well, sequencing a fragment of a 330 kilobase structured with two loops, and so providing the sequence of the fragments. Okay, and this way you don't need the, of course, you don't need the PCR amplification because you are sequencing the original fragments. You don't need the clone amplification because you are sequencing many times the same fragment in a single well, which is nothing more than a reactor sequencing a single DNA molecule. Okay. So what do you do now? So just imagine that you have obtained your sequence reads from a given metagenome sample. So what you can do with the reads, there are two things that you can do. The first one is to assign the reads to the corresponding taxonomy and functionality by an approach that is called direct read mapping. The second thing you can do is to assemble the reads in longer fragments that are called contigs, and the contigs in genomes, if it's possible, and then assign the contigs or the genomes. So the first is called direct read mapping approach, and the second is called assembly-based approach. So let's see how it works, starting from the first approach that is much more easy. So you have just reads. Imagine that we have used Illumina sequencing, and from a given metagenome sample, we have millions of reads, so strands of DNA sequence corresponding to the sequence of each of the millions of fragments generation from the fermentation of the many different genome types. And you need to know which type of bacteria are there and which type of function are there. So you need to assign the reads to the corresponding taxonomy and function. How you can do that by informatics? We will see only few points. So you need to use, of course, the assignment is dependent on annotated database. So you start from available databases with a lot of microbial DNA annotated with the corresponding taxonomy and functionalities. And then you match your reads against them as a query against the annotated databases. So how these databases are constructed and how these databases are used in an efficient way to assign the reads. So the most important database is called Chocoflan, and essentially is made of 99,000 bacterial genomes of high quality, okay? And these 99,000 bacterial genomes belong to 60,000 species, 70,000, okay, doesn't matter. That means that for a given bacterial species, you have more than one annotated genomes belonging to the same species, okay? So now the concept of pangenome. What is the pangenome? The pangenome is the set of gene families that is present in at least one of the genomes belonging to a given species. Just at least one type. So are all the genomes that we can expect to, all the genes we can expect to find in a given species. So for a given species, we have more genome copy, and then the pangenome is represented by the set of genes that is present at least in one of these genome copies representing a given species. So this is the pangenome of a given species, okay? So this way, the first thing that the people creating this assembly read mapping pipeline did is, first of all, collecting the genome of an high quality. Then cluster the genome copy according to the species. So many genome copies corresponding to a given species. Then extracting the pangenomes. So the single genes that are present for each species. Okay, for each species, they were extracting the pangenome. So the single genes that are present at least one time in one genome copy of the genomes belonging to the species. And then for each species, they were, after constructing the pangenome, they were constructing the core genes. The core genes are the gene families present in all the genomes belonging to a given species. So are the genes that all the genomes belonging to a given species possess. Okay, it's different than the pangenome. Okay, and then within the core genes, they created the marker gene set. What means? The marker gene set are the core gene of a given species, so the genes present in all the genome copies of a given species, which are unique of that species. Okay, these are the marker gene of the species. So just repeat the story. So you get 97,000 genomes of an high quality. Then you cluster the genomes according to the species, 60,000 species. So for each species, you have more genome copies, okay? Then create for each species the pangenome. So the list of genes that are present at least one type in the genome, at least one type in the genome, at present in at least one genome of the genome copies belonging to a species. Then create for each species the core gene that are the genes that are present in all the genome copies belonging to a species. Then create the marker genes that are the core genes that are specific of a given species. So not shared with the other species. Okay, these are the three entities. And by doing so, they created what is called the biobakery tool system. So, and this system is what we use for the assignment of the direct quiz. How it works? It's quite easy. So we have our metagenomic reads. Then we match our metagenomic reads against the database, including the marker genes for each species. Okay, so just imagine here. So let's imagine here you have the marker genes of the species 1. Okay. Here you have the marker genes of the species 2. And here the marker genes of the species 3. Okay. So are some genes that are present in every copy of the genomes belonging to the species 1 that are characteristic of the species 1, not present in other species. Okay. Same for the 2 and same for the 3. And then here you have your reads. So your population of 1 million of reads. Okay. Then you match your reads against the databases of marker genes, which are specific for each of the microbial of the 60,000 microbial species. Okay. Then what do you see? Okay, I have one read that match this specific gene, which is specific for the species 1. I have another reads that match these reads, another here, another here, another here. Okay. And then I don't have any reads that match the marker gene specific for the species 2. And I have some reads that match the read specific for the marker gene 3. So now I know that in this metagenome, there is the bacteria 1, the bacteria 3, and there is no the bacteria 2. Okay. Since there are no reads that match the marker genes of the species 2, and there are reads that match the marker gene of the species 1 and 3. Just imagine that you do that with millions of reads that derive from hundreds of different microorganisms, and you match these millions of reads against the marker genes for 60,000 different bacterial species. Okay, and so you can understand which is the abundance of the 60,000 bacterial species in your sample. You can also quantify, because if you have more the species number 1 than the species number 3, you have much more reads pairing to the marker gene of the species number 1 than the reads that pair to the marker gene of species number 3. So for instance, if this microorganism is 10 times more abundant, you would have 10 times more reads pairing here and only few pairing on the reads, the marker gene from the species number 3. And this is proportional to the abundance of the corresponding genomes. So you can also estimate the abundance of the different microorganisms. Okay. So this is just the first step. Okay. Now you know which type of microorganisms are present. So are present the microorganism 1 and the microorganism 3 in your sample. 3 in your sample. So the second step you do is to get the pangenome for the microorganism 1 and the 3. Okay. So all the genes that are present for at least one time in at least one copy of genome belonging to the species 1 and 3. Okay. So then you get the pangenome with all the genes that are present in the species 1, so many. Okay. And then you get 1. And then you get the pangenome from the species 3. You don't need to get the pangenome from the species 2, since you don't have that species. And then you get your reads, and you pair your reads against the pangenome of the species that you know are present in your sample. Okay. And this way you assign the functionality. So you know that there is Bifidobacterium lactis, for instance, and you know all the function corresponding to Bifidobacterium lactis you have in your sample. Okay. And you save time, you save computational time. Since you are pairing your millions of reads only with the pangenomes of the microorganisms you know are in the sample. Okay. And at the end of the story, you still have some reads that do not pair with anything. Okay. You can get these reads not pairing with anything, no with core genes, no with pangenomes, not with anything, and pair only the reads that don't pair with anything, so very few, depending on the sample, of course, but only the one that has not been assigned by using this approach against the annotated huge databases of bacterial genome and so on. But you skip to pair directly your huge population of reads against the huge, super huge metagenome not available in that way. Otherwise, you will spend so much computational time. Okay. So this is a technology that allows to save time and to pair first your reads against highly curated database. And only the reads that are not matched, do not have matched in a highly curated database are paired against the huge databases in which there is quite everything. Okay. You save time and also quality. And this is how you do. So here you find this slide that I will leave to you. You will see many details about how this is done, but more or less the concept is that. So let's see the genome assembly, the other story. So now you have the reads and you pair directly the reads as they are. So the point is that you are assigned fragments of 300 base pair. So it's not so much. So maybe you can create errors since they are short. So maybe it's better to make something longer and to assign bigger fragments of 1 kilobase, for instance, so corresponding to 1 gene. Okay, since of course are much more informative. So the point is that the other approach is not to run directly mapping, but to get your reads and try to assemble the reads in longer fragments. Okay. Theoretically, it's possible to assemble the reads up to fragments longer, up to assemble the reads in their genome of origin. So you can recreate genomes from the reads. Okay. Cos'era? Eri tu, eh? Dormi. So this is the concept of reads assembly. So also it's very easy. So just imagine that you have a single copy of a given genome, okay? Like here, okay? And then you cut the genome in 4 pieces and you have the reads. And then you sequence its reads. How you can put together these reads in the corresponding genome? How you can assemble these reads reconstructing the genome is not possible. You would never know if there is this is the first, the second. How you can is not possible to put together the reads in the genome of origin if you have only one sequence corresponding to each fragment. Okay. So for doing that, you need to have 2 genomes at least, and you need to undergo to random fragmentation. This is why it's important, random fragmentation. So you have the first genome copy, which is fragmented in this way, and the second genome copy of the same strain, which is fragmented in a different way, you see? Then you have all the fragments from the first genome copy and all the fragments generated from the second genome copy. And then you can search for the overlapping of this fragment, okay? By reconstructing the overlapping, you can recreate the sequence of the original fragment up to the original genomes, okay? And this is done by informatically, we won't see that today, but it's done by reconstructing what is called the Eulerian path, okay? That is the theory of genome assembly that results in the construction of what is called the de Bruijn graph. So it's the putting together the different reads in overlapping fragments up to the reconstruction of the longest possible fragments, if possible, up to the reconstruction of the genome of origin. It's not always possible, but this is how it works. So, and the most important result you can get by the assembly is to be able to assemble reads up to the reconstruction of the original genomes. Okay, so starting from the reads, you put together the reads in the genome of origin. And this way, you have a lot of information because this way you have the genome of the microorganism that live in an ecosystem without having isolated any microorganism from the ecosystem. But if you have the entire genome of the microorganisms present in the ecosystem, you know everything about this microorganism, everything. You know the phylogeny, but also you know which kind of genes and function and the metabolism that this microorganism is able to do. And you can create what is called a metabolic model of the microorganism itself. Okay. And by using the metabolic model, you can predict how this microorganism interact with a given molecule or interact with a given system, ecosystem, or interact with a given host. So it's the highest level of information you can get from a native sample. The possibility to reconstruct the single genomes without isolating any microorganism. Since for the microorganism, you can create a model, a metabolic model that represents in silico a kind of twin, in silico twin of the real microorganism that is present in the ecosystem. Okay. So how you do that? It's quite complicated, but there are pipelines. This is a microbiome. You see the different genomes that at different abundances. Then the genomes are fragmented, and you obtain. You go to the sequencing of the fragments. You obtain so many reads, and then you need to put together the reads to reconstruct the single genomes. Okay, and for doing that, you exploit the overlapping between the reads, but there are other approaches that are used, such as the coverage, such as the codon usage, such as the GC%, since you need to find strands of DNA, overlapping DNA, that are very uniform in terms of codon usage, in terms of GC%, in terms of genomic characteristics. Okay, so there are many approaches you can implement for the reconstruction of these genomes. And then when you have the genomes, of course, you can evaluate the quality of the genome. Just imagine that you assemble the reads for a given sample, you obtain, let's say, 50 metagenomes from that sample. And these genomes obtained directly from read assembly for given samples are called metagenomic species. And then you need to evaluate the quality of these genomes. The first thing to do is to check if these genomes are present, the minimal set of genes that are needed for a living organism to live. Okay, so we have a list of genes that must be there. And so you check if that gene are present in your metagenomes, in your genomes reconstructed from the metagenomic reads. And that gene should be present in a single copy. Otherwise, a fake. So there are rules to check for the quality of the genome you have reconstructed by reads assembly. So there is a last technology that is very powerful, but too expensive, in which you have an added sample by using microfluidic tools. You can separate the single cell from an added sample, and then you can sequence the single cells. And then with sequencing the single cells with single genomes, you can reconstruct their genomes. So this is very powerful, since there is not the problem of having different genome types together, and so being able to assign, to reconstruct the different genomes, putting together the reads according to the species of origin, that bioinformatically is not very simple, but it's so expensive. So it costs 10,000 euro for a sample. So while metagenomic sequencing, it costs 200 euro for a sample, this costs 10,000 euros for a sample. So it's still too expensive, and it's not so much useful. Someone invents something much more cheap and usable. So I will turn to the last argument of today. That is the in silico genome mining. So now we know that we can study, well, we know, first of all, that the microbial life on planet lives as microbiome, as microbial community. And we know that this microbial community can provide a lot of functions that are exploitable for many different applications. Okay. And we also know that the first approach for studying this microbial community and for bioprospecting their genetic diversity to find out solution and biotechnological application and biometagenomic sequencing. Okay. And you also know more or less how metagenomic sequencing works. Okay. So theoretically, you have all the pipeline for a given sample, the characterization of the metagenomic community by metagenomic sequencing, and the provision of the annotated metagenomic DNA from that community. So now we have now available thousands of metagenomic DNA from native microbiomes of different kinds being characterized, annotated, and being available in the databases. So, and we can now bioprospect directly these sources, this sequence, this metagenomic sequence, which are available in the databases for finding microbiome-based biotechnology for different applications. Okay. So, and the other very important point is that this metagenomic based biodiscovery is not limited to the cultural microbiome fraction. Okay, so we are discovering directly on the entire functional diversity of the microbial world, not just to the diversity of the cultural fraction as we did up to 50 years ago. Okay. So what we can do? So we have huge databases of metagenomic sequence from different samples, and we can search for novel enzyme, or we can search for novel variants of a known enzyme. So just searching these databases, searching for a selected functionalities. There are a lot of tools, mostly based on machine learning, that have been implemented for scanning the databases, for searching for novel variants of known enzyme, or for novel enzyme for desired function. And when you get it, you can get the sequence, and with the sequence, you can clone the gene, you can express the gene, you can obtain the enzyme, you can also evolve the enzyme, improve the enzyme, expand the substrate range by chemical modification. So there is no limit. You start from nature, and you can also evolve nature. Or you can improve nature just with an engineering modification of the enzyme you get. So there are a lot of chances of improving even the result of the biodiscovery. So, for instance, you can evolve an enzyme by error-prone PCR. So just replicating the enzyme with a PCR that generate error. So you generate randomly some variants of the enzyme, and you check for the functionality to see if there is something that works better. Or you can add a cofactor that allow the enzyme working by photobiocatalysis, so using light for catalyzing the reaction. You can change the catalytic activity of the enzyme, for instance, that's changing a little bit the active site. So there are a lot of chances, and nowadays everything is based on cell-free extraction technology. What means cell-free extraction technology? Means that if you find a sequence of a good gene that may be, according to artificial intelligence, would provide a protein for catalyzing a reaction of interest, of providing a metabolite of interest, you can synthesize the coding sequence by DNA synthesis, and you can express the gene in a cell-free expression system, so without the cell. And then you can evolve the gene just evolving the gene variants by error-prone PCR, expressing all the gene variants and testing for the functionality if there is a variant that works better. So there is a lot of chance for enzyme engineering that can start from the metagenomic bioprospective. So here there are some examples of concrete pharmaceutical, biotherapeutics, biomaterial that has been obtained by implementing this kind of approaches. So starting from the sequence up to the production by biotechnological application of the given enzyme, and then testing for the function and evolving and improving the function. So there is a kind of protein kind of genes that is nowadays the most important for bioprospective application from available metagenome sequence. And these genes are the genes that are responsible for the biosynthesis of what are called secondary bioactive metabolites. Okay, and the genes that are responsible for the production of secondary bioactive metabolites by microorganisms are called biosynthetic gene clusters. And these biosynthetic gene clusters, we will see the structure, are very easy recognizable directly from the metagenome by using genome mining tools, and provide a lot of diversity in terms of metabolites that can be biosynthesized by these genes. Okay. So now we will see the application of biodiscovery on sequence metagenomes, searching for biosynthetic gene clusters, providing new metabolites with different applications in the pharmaceutical sectors mostly, but also as agrochemicals, for instance, or pesticides. Okay, so now we will see a little bit of biodiscovery directly on a specific class of genes providing secondary metabolites that are one of the most interesting targets for the biodiscovery nowadays. So the genes, the biosynthetic gene cluster, are of 4 types. The first one is polyketide synthase. The second one is non-ribosomal peptide synthase. The third one is terpene synthase. And the fourth one is ribosomal post-modified peptide synthase. Okay, so there are 4 types of biosynthetic gene clusters providing this kind of metabolites. They're very interesting. So this is, look at this. This is the polyketide synthase. So the polyketide synthase is a kind of assembly line of different genes which are responsible for the provision of specific secondary metabolites, including antibiotics such as erythromycin and tetracycline, antiparasitic drug, cholesterol-lowering agents, immunosuppressants, and cancer chemotherapies such as the oxorubicin. Okay, how they works? It's very interesting. So these clusters are megaprotein of just imagine some clusters are up to 15 kilobase long that are made of many different models. each module is made of 3 different proteins. Okay, each module with 3 different proteins is responsible for the assembly of a given acyl unit, creating a polyacyl structure that is circularized in a polypeptide. Let's see how they work. Let's see here.
Se viviamo il resto,
Okay, let's see how it works. So, yeah, it is a multimodular protein. Okay, and for instance, you have here just an example. You see 3 modules in line. Okay, each module is made of 3 proteins. Okay, and then you can have more, up to 50 modules. Okay, and which proteins you have in each module? First of all, you have a protein that is called acyl carrier protein. So let's write here ACP. Okay, and the acyl carrier protein carries an acyl unit. This is an acyl unit. Okay, and let's call acyl unit number 1. Okay, and here you have the ACP of the second module. Okay, and then you have the other proteins you have within the module is the acyl transferase that is called AT, and the third is the ketosynthetase, okay, KT. Okay, the same for the third module. So ACP, AT, and KT. Okay, so how it works? So now the ketosynthetase, the acyl transferase, this one, get an acidic unit from the cytoplasm, let's say the acidic unit number 2, and polymerize the acidic unit number 2 over the acyl carrier protein of the second module. Okay, so now you have this. Okay, at that time, the ketosynthetase, this protein here, it's able to get this acidic unit for the upstream module and bind this acidic unit to the acidic unit polymerized in the second module. Okay, and so create this structure. 1. Okay, and then it goes on. You have another acidic unit, the 3, and the ACP of the third module polymerize the acidic unit 3. Okay, just through the action of the acyl transferase that is here. Okay, and then what happened? The ketotransferase of the third module gets the acidic unit from the previous one and polymerize the acidic unit from the previous one up to the acidic unit that has been already polymerized in the third module. And now you have 3, 2, 1. Okay, and you can go on up to 30 modules, each polymerizing 1 acidic unit. And you can create a long acidic chain. Okay, the last module is endowed with a thioesterase, so an additional protein that circularizes the molecule. Okay, so at the end of the story, the line is made circular by a thioesterase that releases the molecules. Okay, But there is something more. Each module, so we have designed 1, 2, 3, and you can have up to 50 models, okay? So many. Each module can be provided by additional enzymes that are responsible for the decoration. So, for instance, ketoreductase, dehydratase, methyltransferase, whatever, that, and can be as it included here within the model. And so, for instance, you have here a dehydratase that works on its polyacidic unit when loaded in the model number 2. And catalyze a modified decoration reaction here. And here you can have a methyltransferase that works on the acidic unit when loaded on the model number 3, in which there is the methyltransferase. So with the addition of additional functional enzyme in each module, you have a decoration of the acidic unit while grow up module by module up to the end. Okay. So you can generate a huge chemical diversity. Okay. Just changing the number of modules and just changing the additional decorating enzyme in each modules and just changing the sequence of the additional decorating enzyme in each modules. Okay, maybe here you have the dehydratase and a methyltransferase, but you make the opposite before a methyltransferase, and then with the dehydratase, you obtain a completely different molecules. So by this system, you can create a huge diversity of metabolites. Okay, since just exploiting the same genetic concept. Here you see exactly how they work. You see many modules. Each one is responsible for the addition of one acidic unit. You see, and here you see step by step the growing of the chain up to the last one that is responsible for the circularization. And each module is also equipped with an additional enzyme. Here you have a dehydratase that works on the acidic unit while loaded on the modules in which the specific enzyme is present. Okay. And consider that each folic acid synthase, so the huge complex, is made of more than 30 modules. Each genes can be different in the number of modules. Each modules can be different in the number of additional enzyme with respect to the middle number of 3, which is present for the decoration. And each gene is different with respect to the sequence of the different modules type, considering the additional enzyme that they have. So you can create a huge complexity of gene with this multimodular architecture. Okay. And why do the bacteria do that? Because with the simple biosynthesis systems, you can create a huge diversity. Just imagine combining differently the different modules, recombination and whatever. You can create linear multimodular proteins of different kinds. So, and from the evolutionary point of view, this is called evolutionary back edge. What does it mean? It means that with a single concept, a microorganism can generate a huge pattern of molecules without knowing which would be the functionalities of these molecules, just generating a chemical diversity. And the microorganisms spend resources for generating a huge chemical diversity. It doesn't know if it works. But the point is, if you generate a huge diversity of functional molecules, maybe there is one that is useful in any condition, even in the condition that you are going to face and you still doesn't know. Okay, this is back edging. So they are bidding on the future. So I generate thousands of molecules. I don't know why. Since maybe there is one of that thousand that now is not usable, but maybe in the future, in future condition, would be important for survival. Okay, this is why the microorganisms generate this huge diversity of molecules, just exploiting this machinery for the biosynthesis of polypeptides. And the second one is non-ribosomal peptide synthase. And non-ribosomal peptide synthase is very similar concept. They provide a lot of antibacterial compounds, penicillin, cephalosporins, anti-tumor drugs, antifungal compounds, generally are all cytotoxic compounds, okay, that can be exploitable, of course, for pharmaceutical application. The non-ribosomal peptide synthase works with a similar concept. So the biosynthesis of polypeptides in nature is not only dependent on ribosome. It's possible to biosynthesize polypeptides also using other systems that are this system. And they work in a very similar way. I don't want to spend so much on describing the details, but it's just imagine this line. Instead of polymerizing acidic unit, they polymerize amino acid. So there is the first module, different enzymes involved. There is the acetylation domain, thiolation domain, condensation domain. So it's different enzymes involved in the biosynthesis system. But anyway, the concept is the same. Since I don't have the board, I don't want to design another assembly line. So just imagine that instead of the acidic unit, you have the specific amino acid, which is loaded by the first module. And then the second module loads the second amino acid. And then there is a protein from the second module that get the amino acid loaded by the first one and connect to the one loaded by the module 2. And then you go on with the polymerization module by module of the given amino acid after the other. And the last module is involved in the circularization. And also in this case, this module is provided with additional enzyme for the decoration of the amino acid, which is a bead in the module in which the enzyme for the decoration are inserted. So the other system is the ribosomally synthesized post-translationally modified peptides. This is more simple. So it's a protein here, normal protein, that is self-catalytic. So you have this is the protein. You have a core part and a leader and a follower. Okay, the leader and the follower can bind over the core. Are two enzymes that bind over the core and modify the core, creating different processes and modifications such as circularization, for instance, and then the two leader and forward are cast by proteolysis reaction. So then this is an autocatalytic system to create chemical diversity. So just you generate the enzyme with the leader and the follower that are able to modify the enzyme itself. Okay. Creating a specific peptide structure that can be responsible of functionalities that are very similar, like the one that are performed by the biosynthetic chip cluster I was talking about before. And then there are terpene. Terpenoids are, of course, a huge class of secondary metabolites, including the cancer, the malaria, steroids, and so on. And they are synthesized from isoprene units that are polymerized, then circularized. It's always the same story. Polymerized, circularized, and modified. Okay. And of course, the enzyme devoted to the polymerization, circularization, and modification are always different and create chemical diversity. So when I say chemical diversity, which is the dimension of the chemical diversity that starts from the genetic diversity? So just imagine that you know what the metagenomic species are. So you get metagenomic reads, then you assemble the metagenomic reads in single metagenomes, and you have a metagenomic species. Okay, we were talking about that before. And this is a study that's been carried out a few years ago, from which they got 10,000 metagenomes from diverse habitats. And you see here, you have metagenomes from marine ecosystem, from terrestrial ecosystem, from host-associated ecosystem, and from engineered, so anthropic ecosystems. So up to 10,000 different sample, metagenome sample, from which the sequence have been already obtained and released in public databases. Okay, so they get the samples and then they reanalyze these samples, try to de novo assembly, so put together the reads and assemble de novo the metagenomic species. Okay, to assemble genomes from metagenome that were already provided and available in the database. They have been successful in assembling 52,000 metagenomic species, okay, of which almost 10,000 were of innate quality, meaning the genomes were quite complete, okay. And so they were asking, which is the novelties of this diversity, this microbial diversity, these genomes we have been able to assemble from metagenomic reads with respect of what we already know in terms of microbial diversity. So what they did was simple. So they got all the already characterized microbial genomes available in the databases. So I'm speaking about 200,000 genomes from isolates, 300,000 genomes from metagenomes that were already present in the database, 2,000 genomes from single cells, okay? And they were combining all the available genomes with the old genomes they have newly generated by generating the metagenomic species from the 10,000 metagenomes available in the databases. And so they were putting everything together, and these genomes were belonging to 30,000 different microbial species altogether. Over the 40,000 different microbial species, 45,000 different microbial species, 12,000 were candidate new species and were provided by their study. So, essentially, in few words, by assembling metagenome available in the databases in metagenomic species, they have been able to discover 12,000 new microbial species, not known in 2022. Okay. And then they explored the taxonomy of these 10,000 new bacterial species, and they were distributed in all the phylogenetic branches. So the taxonomy was so wide, since from so many different ecosystems. And if you look at this diversity, the green line represents the microorganisms that have been isolated. The other one, the gray one, represents genomes of microorganisms that has been obtained by metagenomic species. So you see the great diversity, microbial diversity we are aware today is of bacterial genomes from microorganisms never being isolated before. Okay. So directly reconstructed from metagenomic samples. So turning to secondary metabolites, then they were searching for secondary metabolites gene cluster in these 12,000 new microbial species they have been able to find and detect in terms of reconstructed microbial genomes from metagenomics, so never isolated. So essentially, they have 44,000 genes, including non-ribosomal peptide synthase and polyketide synthase. They have 32,000 terpene gene cluster. There are 12,000 ribosomal processed peptides, and of these, 87,000 of these biosynthetic gene clusters were completely new. So potentially they code for molecule we don't know anything. And since the biosynthetic gene cluster, by definition, provide molecules which are bioactive and which are interesting in terms of occultation, since they are pharmaceutical, for instance, potentially this is 80,000 genes coding for secondary metabolites with possible bioactivity in terms of antimicrobial, new antimicrobial, immunostimulants, immunoregulators, or cytotoxic compounds to be used as anti-cancers. Okay, so it's very important discovery action. So let's see some databases. And this is important because this is part of your own work. So here there is the biosynthetic gene cluster atlas. The biosynthetic gene cluster atlas is a database of 1 million of gene clusters, all geolocalized. So you know the gene cluster, and you know where it has been found, and you know if it's code for something. Okay. And this is how the database was constructed. It's from 2024, so very recent. And what is important is that the biosynthetic gene cluster include all the regulatory areas. So if you clone the gene, there are all the systems for controlling the expression of the gene. And of course, the obtaining of the protein and the metabolites. So they are substantive. And associating the biosynthetic gene cluster with the biogeography, they have been able to understand something about the functionalities of the biosynthetic gene cluster in terms of evolution and ecosystem interaction. Okay, so biosynthetic gene cluster, biogeography means connecting the biosynthetic gene cluster with the ecosystem features, and so speculating how the biosynthetic gene cluster, why that biosynthetic cluster is evolved for the specific ecosystem. Okay. And there is another dedicated database to the, this is a publication from Nature, to the biosynthetic gene cluster, specifically from marine origin. And there is 40,000 biosynthetic gene cluster that have been detected across the ocean. You see the dot correspond to the biosynthetic gene cluster detection sites. And also here they will start to understand if there were connection between the specific functionalities coded by biosynthetic gene cluster and the ecosystem. So of course they are exploitable since they are bioactive molecules such as cytotoxic compounds. But why the microorganism synthesize this molecule? Why they need that? Why they make this happen? Why they maintain such huge diversity, such huge chemical diversity. And so they were trying to connect these things, and they were found that some biosynthetic gene cluster coding, for instance, for carotenoids and polyenes, or real polyenes, or siderophores were essentially present in any marine ecosystem. So are the biosynthetic gene cluster coding for molecules that are needed to live in the ocean. But there are some other biosynthetic gene cluster that are enriched in prokaryotic enriched community, and it's just the terpene. Maybe that biosynthetic gene cluster code for molecules which are important for competition between different species, because they are present, which there are in ecosystem which there are so many bacterial species, and that there are biosynthetic gene clusters that are present in ecosystems that are virus enriched. So maybe are the biosynthetic gene cluster important for defending the microorganisms against the virus. So that to say, matching the chemical diversity with the ecosystem diversity, we are trying to understand which is the function of these biosynthetic gene clusters for the microbial life, and how they can control for microbial processes. So then, of course, there are microorganisms that from the sea that are particularly enriched in biosynthetic gene cluster. One is cyanobacteria, and Myxococcota surely is very rich. Rhodococcus and Plantomyceota are all bacteria that are characterized by having a high social behavior. So somehow this biosynthetic gene cluster are important for competition, are important for competition interaction between different species, but also are somehow involved in the social behavior. So microorganisms that are much more social, so are much more interacting between each other, are much more enriched in biosynthetic gene clusters. So this is another very recent publication about the potentiality to exploit this huge dynamic diversity coding for chemical diversity for, of course, biotechnological application products such as antimicrobial, anti-inflammatory pesticides, and not toxic phytochemicals. And what I want to point out that over the 80,000 biosynthetic gene clusters that I'm considering that they are the one of marine origin, the 3% has no function. So mostly we know that they are polyketides, non-ribosomal synthase, since they have this kind of modular structure. But we don't know, we are not able to predict the metabolites that are going to synthesize. And so there is a lot of research now trying to ameliorate our bioinformatics system for assign metabolites to the biosynthetic gene cluster and for predicting the functionalities to the metabolites. So only to put in line the ecosystem, the biosynthetic gene cluster, the metabolites, and the function. And then for the one which are interesting, we can clone the gene, express the gene, obtain the metabolites, and test in functional, in vitro or in system for the corresponding functional activity. So this is how it works. Essentially, there are two approaches. One is, first of all, you must consider that the biosynthetic gene cluster possesses all the genes for its own regulation. So essentially, starting from the metagenomic fragment, you can obtain the expression of the gene in an heterologous system. So you get the gene, you put the gene in a specific bacteria or in a cell-free expression system, and you obtain the protein. And from the protein, the metabolites. Okay. So what is done now is using that peculiarity of biosynthetic gene cluster for creating some biosynthetic gene cluster capturing system. So you get the metagenome, you cut the metagenome, you let this huge, now you cut the metagenome in huge fragments of 50 kilobases. Okay, so very big, not sure. And then you cut the metagenome in big pieces, and then you clone each pieces in a specific expression vector that is functional for the expression of the protein, the pieces code, and for obtaining the metabolites and for testing functionalities. Okay, and this is something that is doable nowadays, and it's another powerful approach that's starting from DNA, how to test DNA for the biosynthesis of biosynthetic gene cluster, and then how to obtain the metabolites that are produced by the biosynthetic gene cluster and test the metabolites for the functionalities. So here there are other tools that are so important for the virtual biodiscovery. This one is MetaSeq. This is a database in which you can upload here the name of a metabolites you want, and the database provides you the corresponding gene, the pathway, and the microorganism that is possess the pathway. So starting from the metabolites you want, you can know the microorganism that is able to synthesize the metabolites. Always based on microbial genomes. Then there is another database that is very important. It's called BackDive. That is the opposite. You start with the microorganism, and the database inform you. Just type the name here, and the database inform you about the taxonomy, the physiology of the microorganism, the metabolism, the enzyme that the microorganism possess, the ecology, where it's been isolated, and how, and also how you can grow the microorganism, so the grow media and the grow condition. Okay. So the first database, you want a molecule. We'd like to know which microorganism is able to provide the molecule. This second database, you need the microorganism inform you about the biology of the microorganism. That's also most important, how you can grow this microorganism and the genomic feature of this microorganism. Okay. So by crossing the two databases, you can know everything about the microorganisms that are already known. And this is the most important one. Know a microorganism. These databases is able to provide you information about the biosynthetic gene cluster the microorganisms possess and what the biosynthetic gene cluster are predicted to synthesize. So which kind of metabolites you can get. Okay. How it works? It works just uploading in the database a code that corresponds to the genome sequence of a given microorganism. So you have microorganism, you have the genome, you have a code that connects the microorganism to its genome. You upload the code in this database, and the database will predict all the biosynthetic gene cluster the microorganisms have, and also the products these biosynthetic gene cluster are able to provide. Okay. So, last point, Nagoya Protocol. So we are speaking about biodiscovery. So we have said many things in a few times. Anyway, we are speaking about biodiscovery. That means that you find a microorganism that is able to provide the bioresources such as a new antimicrobial compounds, and you can get money from this resource. Okay, maybe you are a company, you have a big pharma company. So let's imagine that you are a company from the United States, and you get this microorganism that produce a new biosynthetic gene cluster that has functionalities as a new antibiotics, so you can get a lot of money. You get the microorganisms from the South Africa. So maybe the South Africa should get something as well from the commercialization of the bioresources, since the owner is the South Africa. So when you find a bioresources of any time, if you want to exploit the bioresources everywhere, you need to pay the country of origin. And this is possible only since 2012. So the Nagoya Protocol, now 2014. Okay. So everything was isolated before the 2014, is not within the Nagoya Protocol, and you can do what you want. Everything is isolated after the 2014 is within the Nagoya Protocol. So if you want to use it, you need to provide benefit also to the country for which the microorganism has been isolated. Okay, so it's very important. Of course, the United States is going to exit also from this one. It's very complicated, something like lawyer. So then there is bidding and so on, and then deciding this is the percentage. So you need to find agreement. So the bioresources is your, and I want to explore the bioresources, and there is bidding. So I give you 5%. Is it okay? No, I want 10. Okay, 8, something like that. But the ownership in terms of provenance of the biological diversity needs to be recognized. It's correct. So here, some marine ecosystem or allobiomes from which the biodiscovery of biosynthetic gene cluster has been historically carried out in the past. Metazoan, spongy, cnidarians. That's, of course, live in association with the complex microbial community that essentially defend the host. So they're responsible of the host defense, and of course they get nutrients from the host, and in response they defend the host from pathogens, and so providing secondary metabolites which are cytotoxic. And since they are cytotoxics, are also anti-inflammatory, anti-cancer, anti-pesticides, and whatever, anti-life, let's say. Okay, so it's very easy to understand why we can find a lot of important bioresources in terms of secondary metabolites in metazoa, spongy, and cnidarians. And also microalgae and seagrasses are resources of secondary metabolites. In that case, microalgae and seagrasses are responsible for the biosynthesis of their own secondary metabolites. And of course, fungi, it's capable to biosynthesize so many secondary metabolites, and also free-living microorganisms, bacteria, archaebacteria, also by their own synthesized secondary metabolites, as we are seeing, that are responsible for the interaction between different species, different microbes with the ecosystem, for the bacterial sociality, and so on. And so for what you can use, the application is agronomy, of course. There are a lot of applications in agronomy, but also the most important one are in the healthcare. Here you have all the drugs that are secondary metabolites of marine microbiome origin already approved in clinical trials for treating cancers. So you see how many they are. So now your homework. First of all, so you are asked to run, as I was saying, a kind of biodiscovery experience by yourself. So first of all, you need to select a marine ecosystem. You believe can be somehow of interest for running a biodiscovery application. For selecting the marine ecosystem, you can use these tools, in which you can see which are the marine ecosystem mostly enriched of biosynthetic gene cluster. Or you can select any marine ecosystem you like for any region, okay, as you like. If you want to select this Adriatic Sea, Northwestern, you can select the Northwestern Adriatic Sea, no problem. Okay, so you can use this tool if you want to find a marine ecosystem which is enriched in biosynthetic gene clusters, or microorganisms enriched in biosynthetic gene clusters, or can select any marine ecosystem of interest you like, the one you are studying, what you want. And then, come back. From the marine ecosystem, of course, you can select, as you like, a microorganism that lives in the marine ecosystem you are interested in. So it's easy, it's just a literature research. So just sediment in the Northwestern Adriatic Sea, just collect some papers and see which microorganisms live there. So there are many papers, just report the microbial diversity of any ecosystem, okay? Then select one. This is your choice. You are free to select the microorganisms you like. Okay. You can select also more. Okay. And then from this microorganism, you need to provide the ecological and the metabolic information by using the tool BacDive. You remember BacDive. And then you have the ecosystem from which you can isolate the microorganism. You have the microorganism. You have the metabolic information of the microorganism. Okay. And then at first step, you need to get the genomic DNA of the microorganism. Okay. When you have the genomic DNA of the microorganism, you need to upload the genomic DNA of the microorganism, and you need to bioprospect the genomic DNA of microorganism to understand which kind of secondary metabolite gene cluster the microorganisms possess and what they code for. You can do that with different microorganisms up you find one that provides something that is of interest for you. Okay. At the end of the story, you need to prepare a very short research project in which you say the taxa, where you expect the microorganism, where you expect to isolate the microorganism, which are the genomic and metabolic feature of the microorganism, which secondary metabolites these microorganisms can provide, and which are the possible application for these secondary metabolites, pharmaceutical, antimicrobial, anti-cancer, whatever. Okay. So, and you can do that by using these tools. So just imagine that you are working in a given ecosystem. Then you go on the literature and you search for the microbial life from this ecosystem. If you're working with a fish, you can search for the microbial life in the gut of the fish you're working with, or plant, or whatever. Okay, you will find that for sure. Then you use the back dive, and by using the back dive, you type here the name of the microorganism, okay, and you will get all the information of this micro. See here I was typing the name of Paraphotobacterium marinum, just as example, and here it provides you as a number, mesophilic, gram-negative, and forms circular colonies. And here you have all the information about taxonomy, and here you have the gram strain, the cell lines, the cell weight, the cell shape, the motility, the colony size, colony shape, incubation period, cultivation medium. And then you go on. This is just a part of the web pages. You have information about genomic and isolation, ecological feature, and whatever. Okay. And then with the name of this microorganism, so just imagine you have Photobacterium marinum, you go on this website that is the National Library of Medicine, the NIH, okay, and here you see here's the NCBI website, okay, this is the address. Here you just there is a menu in which you need to highlight genome. Okay, you type here the name of the microorganism, and then you run search, and then you will get a page in which you will have all the genomes corresponding to the microorganism. Then you select the one with the mark in green, like this. Since are the high quality genomes, you will have many, but you will just select this, just type over the one with the mark in green, and you will have a page describing the specific genome belonging to a Photobacterium marinum. Okay, so starting from the beginning, you type the name, you write genome here, you have search, you will have many genomes from Photobacterium marinum. Okay, then you select the one with the green spot, then you click over the one of the green spot, and you have a second page in which you have all the sequence corresponding to that genome type, to that species, okay? And then you need just to copy that code. That is the code corresponding to a genome type corresponding to your species, okay? And then you paste here on anti-smash, the tool for the prediction of the secondary metabolites, the code. And then you space submit. Okay. And this is the code of one genome of Photobacterium. And then it takes a little, few minutes, maybe also an hour, depending on don't know what. And anyway, after the end of the elaboration, you will get this page, okay, with the status done. And here there is the word result. It's very small. You click over this result here, okay, and you have a page here with all the secondary metabolites gene clusters that are present in the genome of the microorganism you have selected. Okay. And here you can see the functionalities of the gene clusters. Okay. Of course, there are many. This is just a page, a screenshot of the beginning of the page. You, based on the gene cluster you have, you need to see if there is something that is predicted to code for metabolites of non-metabolites. Okay. And you sign the name of the metabolites, you go on the website and search for the functionality. Okay. And this way you have your ecosystem, a microorganism living in an ecosystem. You know how these microorganisms can be isolated and how can be grown in the lab. You know the genome of the microorganism, and you know the biosynthetic gene cluster the microorganism can code for, and you know if there are some biosynthetic gene clusters that code for product of potential application as what you like. Okay. You can repeat this, it takes only a few minutes. You can, for instance, if you go to your ecosystem of interest, you can find many bacteria, you can reiterate this pipeline with all the bacteria up to find something that is of interest in terms of product from its biosynthetic gene cluster. So just not limiting to one microorganism. So you can take a little bit more time and run the same pipeline for many bacteria just you find in a given ecosystem. Okay. And this way, after the end of the story, you can prepare a kind of paper in which you say, okay, from this microbiome, isolate this microorganism that is able to produce a biosynthetic gene cluster can be imported as a new antimicrobial compound to be used in aquaculture. Okay. So just put together everything in a short story, and it would be original because it would be the result of your biodiscovery. So just imagine that what you have done could theoretically be done for the first time. Since, for instance, you have the microorganism Photobacterium XXX, okay, that has been isolated from fauna. Okay, and the people who have isolated this microorganism has provided the sequence of the corresponding genome for doing something. I don't know what. Okay, but maybe in the paper that isolated the microorganism, sequenced the genomes, and provided the sequence of the genome, they had not been interested in biosynthetic gene cluster. Or maybe they did that sequencing before anti-smash was available. So we are speaking on, see how many bacteria there are. So we are speaking 200,000 genomes of microorganisms, even more, 10,000, hundreds of thousands. So that is possible. So this would be you, for the first time, exploring this genome for the biosynthesis of secondary metabolites by using anti-smash. So you can create something original, never done before, even using data already available, but you are using data already available, genomic data already available for different purpose and by using different tools with respect to the ones have been considered when the sequence, the genome sequence was provided. Okay. So I think we can close now. Now I'm open to any question. If you want to try the tape line, you can come and try. Some of you, Daniela. Eh? Ah, è facile. Come. Try with another number. You can do here. Works here. So the other guy can see. So just go on CBI. Write your telephone number. Una volta qualcuno lo usa.
Tutto no.
Ok. Okay, genomes. Go slowly. You see in CBI genomes. Now you type the name of the microorganisms you like. Libro, qualche libro,
Sì.
un libro cattivo.
Io avevo cercato i libri originanti questi. Aspetta, mi vengo.
Dov'è?
Il Gino, aspetta, mi ricordo. Il Gino. Questo. C'è un 12 mi sembra. Però perché non lo scrivo.
Non ricorderò mai. Okay, here you get the you see here you have many so strains, sorry, belonging to that species. Of course, there are more strains for a given species. Okay, so the database are huge, and you select the first one. No, just click the red one. Okay. Okay, now you say, okay, I select this strain, the first one, and you click, and you have the genome of that strain belonging to the species you have selected. Go down. Genome features. Just copy this number. Yeah. And then you go to anti-smash. Okay, now you type the number, submit bacteria. Okay. Okay. Okay, now submit and wait. Yeah, maybe it takes some 1 hour. Anyway, this way is the right way. So it can take 10 minutes or 1 hour. Done. Okay, result is very poor of biosynthetic Vibrio ferrin. So there is region 1 that we get. So here you have the name of the biosynthetic gene cluster, the structure, and the biosynthesis of this Vibrio ferrin. That actually I don't know what it is, but anyway you can search what is ferrin. Should be something for siderophores, something like that, capturing iron that generally pathogens needs. Iron, so metals, so they like it. So anyway, this is how you can do. Okay, so it's very easy, and you can test different microorganisms up you find something you like. Okay, then you create a folder. Then you put together all your report in a folder. And you say, wait, Marco, the folder is full, and I will go to the folder and check all your report. Don't send the report to me by email. Okay. Otherwise, they would get lost, for sure. Okay. So just you can organize by yourself and share the folders. Share a folder, and then when there are all the reports, you say to me, and I'll check all. Okay. Okay. Beautiful. Let's see.